Hello, grade six. This is your science summary, your first science summary, and it's about the organization of living at the microscopic level. Throughout this period, students will be able to identify the basic unit of structure and function of living things, conclude that the microscope is a tool used to observe very small objects which cannot be seen in the naked eye, and recognize and label parts of the microscope and know the procedure of using a microscope. So the first thing we looked at was cells, okay? And the most important thing that you need to know about cells is that all living things are made out of cells, okay? Please, it's really important. The thing has to be living. What do I mean by living? Is alive, okay? For example, humans have cells, plants have cells, animals have cells, okay? Plastic, because it's not living, okay, does not have cells, all right? So please, this is really important. Living things have cells. Wool, which is found on a sheep, has cells, okay? Even if, for example, a table made out of wood, okay, this wood will have cells, okay, so it has to be living. Leather, okay, for example, is also made out of cells, okay. This is really important for you to understand. All living things are made out of cells. How do we see cells? We use a microscope because our eye cannot see small objects such as cells, so therefore we have to use a microscope. This is how we label a microscope. First, we have a tube which is connected to the ocular. Okay, so the ocular is what we see with. We've got the revolving nose piece. The revolving nose piece revolves, so it goes round, so we can change the objective. Then we have the stage. The stage is where we put our specimen, the thing that we want to see on it, and it's held by stage clips. So then we have the condenser. Okay, so the condenser controls the amount of light that goes in through the specimen. We've got the light source. So the light source is we need light to see our specimen properly. The base is what holds everything together. The base and the arm, here's the arm and here's the base, basically as part of the structure of the microscope. Then we have the coarse focus. Remember, the coarse focus brings the stage up, okay? It brings the stage up and down. And then the fine focus, remember we said that when we look under the microscope, once we do the stage focus, the slide will be a bit blurry, so we have to focus it with the fine focus. How to use a microscope. First, we have to eliminate the microscope by turning on the light source. Usually when you plug in the microscope, the light source will come on by itself. We place a microscope specimen on the stage. So the thing that you want to see, you put it on the stage and you hold it using the stage clips. Then you adjust the objective on the lowest magnification. So there's the objective, and you just turn around the revolving nose piece so you can get the lowest objective. Then you've got the coarse adjustment knob, and this reduces the working distance between the slide and the objective. So basically what it does is it brings the stage up to the objective so that you can see it clearer. You then place your eye on the eyepiece. When you place your eye on the eyepiece, it's going to be a bit blurry, so you have to use the fine adjustment knob to get the optimal tuning point so that you just make it a bit more clearer. And then finally explore the specimen. So throughout this lesson, students are able to now identify the basic unit of structure and function of living things. Remember, we said that cells, okay? That cells are the unit of structure and function of living things. We concluded that the microscope is used to observe very small objects which cannot be seen by the naked eye, and we saw that in the lab. Remember when we went and we saw the cells under the microscope? Recognize and label parts of the microscope and know the procedure of using a microscope. So we saw in previous slides how to label the microscope. We looked at the stage clips, the ocular, the revolving nose piece, the coarse adjustment knob, the fine adjustment knob, the base. Okay, so all of these are parts of the microscope. And the procedure of using the microscope, remember, is we have to turn it on, and then we have to put the specimen onto the stage. We then have to put the objective on the lowest magnification. Then we have to use the coarse adjustment knob to bring the stage up to the objective. And then we see the specimen through the ocular. Remember, it's going to be a bit blurry, so we use the fine adjustment knob to make it clearer, and then we explore the specimen. So that's like kind of a nice summary about this period.